Hey everyone, welcome to another deep dive. You know how much we all love philosophy, and I think today's going to be a really interesting one.
Looking forward to it.
We're going to be looking at the history of the gene concept, and like all things in science and philosophy, it's not quite as straightforward as you might think.
Well, it rarely is it?
Right, exactly. So we're using an entry from the Stanford Encyclopedia of Philosophy as our source material for this deep dive. And just to get us started, the idea of a gene has changed a lot over time, you
Oh,
know?
absolutely.
Like it's really evolved alongside all the major changes in biology, which makes sense.
Yeah, I mean, it's like it's gone from, just thinking about cells and molecules to like fundamental concepts.
Totally. So basically ready to unpack the philosophical side of genetics.
Let's do it.
Okay, so to start us off, we got to go all the way back to the 19th century with Gregor Mendel,
Way back.
right? Like way back. He was a monk who was super into heredity.
Okay, a monk.
Yeah, you wouldn't think a monk would be like a pioneering scientist.
That's a good point.
But Mendel's work with pea plants was revolutionary.
What was so special about it?
Well, he wasn't trying to make like some huge theory of inheritance or anything, but his experiments,
Yeah.
which he did super carefully,
Yeah,
they showed some really interesting patterns.
I see.
And actually what's so fascinating about Mendel is how like systematic he was.
Oh, really?
Yeah, especially for that time period. He's like, okay, I'm going to look at specific traits, like just the color of the flower or the shape of the seed. And then he'd use statistics to really analyze what he was seeing.
Interesting.
Oh, and he even came up with his own way of writing down his findings, like a whole notation system.
Wow, he was ahead of his time.
Right. It was
Yeah.
totally different from what other biologists were doing that.
Yeah.
It's way more rigorous.
Much more scientific.
Exactly.
Ah.
But, okay, so get this. Mendel did all this amazing work, and basically no one noticed for decades.
You're kidding.
I know, it's crazy. It's like someone hid a scientific time capsule, and then it was finally rediscovered, like, bam.
And that was when everyone started paying attention.
Right? In 1900, it totally revolutionized biology.
I can imagine.
But, okay, so I know it's been a while since high school biology for me.
Me too.
So, like, can we just break it down? What exactly did Mendel find, those patterns he saw? What were they?
So basically, he realized that traits in these pea plants weren't just like mixing together.
Right.
Like they were passed down in predictable ways. For example, he crossed like a purebred purple flowered plant with a purebred white flowered one, right?
Okay.
All of the kids, the offspring, they all had purple flowers.
So purple was dominant.
Exactly. But then he crossed those offspring, and what do you think happened?
I don't know what happened.
Some of them had white flowers again.
Oh, so it came back.
Yeah, it came back, and it showed up in like a specific ratio. So it wasn't just random.
Huh. So there had to be something else going on underneath, like not just the mixing of the colors.
Right. It meant there were like units of inheritance being passed down from the parents.
Oh.
Mendel called them factors. Yeah, and they acted like little particles, being passed from one generation to the next. It was a huge deal at the time.
So that's where the idea of a gene started.
Pretty much. It laid the groundwork for the whole gene concept.
Okay, but how did we get from factors to genes? Like what happened there?
Well, it took some time. Scientists were trying to figure out exactly what these units of inheritance were. They were using all sorts of terms like unit characters and allelomorphs, but those were often attached to like specific theories about how it all worked.
Oh, so it was a lot to keep track of.
Exactly. They needed a word that was like precise, but could also change as they learned more.
Right. Makes sense.
And then finally in 1909, Wilhelm Johansen, who was this Danish botanist, came up with the word gene.
Gene.
Yeah, he wanted a term that didn't have all that extra baggage from the old theories.
I see.
So it was kind of like a placeholder for something really important. They knew it was there, but they didn't know exactly what it was or how it worked.
So at this point, genes were still just like a concept.
Yeah, they were abstract. They were understood by what they did, like how they affected what you could see, but no one knew what they were made of.
It's so interesting how one word can be so important to science.
It really is. And that brings us to the next big step. Thomas Hunt Morgan and his team.
Ah, yes, the famous fruit flies.
Those little guys. They were mapping genes onto chromosomes.
Right, so they were like the first ones to connect those two things.
Exactly. They noticed that certain traits tended to be inherited together,
Okay.
which meant that whatever was causing those traits had to be physically close together on the chromosomes.
Oh, like linked.
Exactly.
Yeah.
And by looking at how often things were linked, they could actually map where the genes were.
Wow, so they're making a map of the genome like way before DNA sequencing.
Exactly. It was huge. Now the abstract idea of a gene was connected to something real.
Must have changed everything.
It did. Now they had a physical basis for the gene.
So it wasn't just a theory anymore.
Nope, it was real. And that was a big deal for genetics.
Mm-hmm.
But even with this discovery, there were still a ton of questions left.
Like what? Well, like what are genes actually made of? And how do they make those traits appear?
Right, big question.
And those questions would lead to even more discoveries about the molecular side of genes.
Mm-hmm.
And one of the big things they found was this connection between genes and enzymes.
Enzymes.
Yeah. You know, those things that make all the chemical reactions in a cell happen?
Right, so we're going from like the structure of chromosomes to what they actually do.
Exactly.
Right.
Researchers started to think about how genes worked in a similar way to enzymes, you know, with this chemical reaction.
Okay.
And then they got really into the idea of mutations.
Mutations.
Yeah, like what happens when a gene changes? They had scientists like Herman Miller looking at what X-rays did to genes.
So they were using radiation.
They were. It was like they were trying to mess with the genes on purpose.
Wow.
And they found out that X-rays could actually change genes and make traits show up differently.
So that meant genes were like physical things you could actually change.
Right, exactly.
Yeah.
Not just some abstract concept.
Yeah.
And that leads us to something really cool. The link between genes and enzymes.
Okay.
This is where we get the one gene, one enzyme hypothesis.
Oh, yeah, I've heard of that.
Yeah.
But wasn't it kind of controversial?
Well, yeah, it got more complicated later on, but it was a big step forward.
So what was it all about?
So it was all about figuring out how genes actually make physical traits, you know?
Right.
Like if you have the gene for brown eyes, how does that actually make your eyes brown?
Right.
And these two guys, George Beadle and Edward Tatum, they used bread mold to study this.
Bread mold.
Yeah, bread mold. It's
Yeah.
really good for genetics research.
Yeah.
And they found out that if a gene got messed up, it could break a specific enzyme.
Oh, so like one gene controlled one enzyme.
Exactly. And if that enzyme was broken, it could change a whole metabolic pathway in the mold.
So it wasn't just about the gene itself, it was about the whole chain reaction it caused.
Exactly. They were connecting genes to the actual chemistry happening inside the cell.
That's pretty amazing.
It was. But you're right, it did get more complicated later on. The one gene, one enzyme idea wasn't always true.
Okay, so what happened?
Well, we need to talk about the elephant in the room, you know?
Let's say.
We've talked about genes and chromosomes and molecules, but what are genes actually made of?
Oh, right. We haven't gotten to that yet.
And this is where things get really exciting, because that's when scientists figured out that DNA was the genetic material.
DNA.
Yeah, the big one. This was a huge turning point in biology.
So who figured that out?
Well, the stars of the show were James Watson and Francis Crick.
Oh, yeah. They're like super famous.
They are. Their story is legendary. They're the ones who discovered the double helix structure of DNA.
The double helix.
Yeah, and that changed everything.
How so?
Well, their model showed how DNA was structured, you know, that elegant double helix shape.
Okay.
But it also gave us a clue about how DNA copies itself.
Oh, so you mean like how genetic information gets passed down?
Exactly. Each strand of the DNA acts like a template to make a new strand. It was brilliant.
So the structure of DNA itself told us something about how it worked.
Exactly. And once DNA was the main focus, everyone wanted to know how genes actually did their job, you know?
Right, like now we knew genes were made of DNA, but how did that make proteins?
Right, because proteins are the ones that actually do things in the cell.
Okay.
And there was this guy, Seymour Benzer. He was doing some amazing work.
What was he doing?
He was looking at mutations, but like really, really closely. He was mapping mutations within a single gene.
Wow, so he was like zooming in on the gene.
Exactly. And what he found supported the idea that DNA was the genetic material,
Wow, so
because he showed that even a single base in the DNA could change a gene.
it was like those bases were the building blocks of heredity.
Exactly. And around the same time, they were working on the genetic code.
What's that?
So the genetic code is how the sequence of those bases in the DNA tells the cell how to make a protein.
Oh, so it's like translating from one language to another.
Exactly. The language of DNA to the language of proteins. And it was Francis Crick again who came up with this idea. He called it the central dogma of molecular biology.
The central dogma.
Yeah, it's like the basic rule of how genetic information flows.
So what is it?
It says that DNA makes RNA and RNA makes protein.
Okay, so DNA is like the blueprint, RNA is the messenger, and protein is the final product.
Exactly. That's a great way to put it. And even though we've learned more since then, the central dogma is still really important in biology.
But I'm guessing things got more complicated.
They always do, right?
Yeah, like biology's never that simple.
You're right. So what happened?
I don't know, tell me.
Well, scientists started to realize that genes weren't always just a straight line of DNA.
How do you mean?
They found these things called split genes.
Split genes.
Yeah, where there would be these non-coding regions in the middle of the gene.
Non-coding.
Yeah, they're called introns, and they interrupt the coding regions, the exons.
So a gene could be like broken up into pieces.
Exactly. It's like having a sentence with a bunch of extra words in the middle.
That makes things a lot harder to understand.
It does, and it gets even weirder.
Oh,
Well,
yeah.
they found out that those exons, those coding pieces, could be put together in different ways.
Like mixed and matched.
Yeah, it's called alternative splicing.
Alternative splicing.
Yeah, and that means one gene can actually make multiple proteins.
So it's not just one gene, one enzyme anymore.
Nope. It's way more complex than that.
Wow, biology is so cool.
It is. And on top of that, they found a ton of DNA that didn't code for proteins at all.
Really?
Yeah, tons of it. They used to call it junk DNA.
Junk DNA.
But it turns out it's not junk at all.
What does it do then?
Well, it does a lot of things. It's involved in regulating how genes are expressed, turning them on and off.
So it's like the control panel for the genes.
Exactly. And with all these discoveries, the idea of what a gene is became a lot more complicated.
So there wasn't just one definition anymore.
Nope, there were all these different gene concepts, each one focused on a different aspect of how genes work.
It's like the gene itself became a puzzle.
And that puzzle takes us into the world of modern genomics and systems biology.
So what happens there?
Well, that's where we start to see just how dynamic the concept of a gene really is.
So much to think about. I mean, we've come a long way from Mendel and his pea plants to like split genes and all this non-coding DNA.
We have, and it just keeps getting more complex.
Right. Like, see, it seems like the more we learn about genes, the less we actually know.
Well, that's how science works sometimes. The more you discover, the more you realize how much more there is to discover.
True. But you mentioned this thing called high throughput technologies.
Oh, yeah.
Like, what does that even mean?
Well, it basically means we can now analyze entire genomes really quickly. We can get huge amounts of data.
So it's like big data, but for genes.
Exactly. Big data genomics.
And that must have changed things a lot.
Oh, it has, completely. It's given us so much more information about how genes are regulated and how they interact with each other. It's incredible.
Can you give me an example?
Sure. So there's this project called ENCODE. Yeah, it was this huge effort to figure out what all the parts of the human genome actually do.
Okay.
And it turns out there's a lot going on. Like transcripts can overlap, and you can have functional products from DNA sequences that are super far apart, and regulatory sequences are everywhere.
Wow. So it's not as simple as like one gene, one spot on the chromosome.
Not at all. It's way more messy and interconnected than we thought.
So what does that mean for the concept of a gene?
Well, it means we need to be more flexible. There's no one definition of a gene that works for everything.
Okay.
It depends on what you're studying and what questions you're asking.
So in a way, the gene itself has become like a philosophical problem.
It really has. It makes you think about how we define things in biology. What makes a gene a gene?
Well,
What are the boundaries?
it's like the gene is an idea that keeps changing.
Exactly. And philosophers of science are all over this. They're debating whether the gene concept is evolving or if it's gone through these radical shifts over time.
So some people think we're still talking about the same basic thing, just with more detail.
Right. And others think that different gene concepts are actually talking about totally different things.
It sounds like a really interesting debate.
It is. It shows how important philosophy is for science. We need to think critically about the concepts we use, especially when we're making so many new discoveries.
I agree. This whole deep dive has been a real eye-opener.
It's amazing to see how far we've come from those early days with Mendel's pea plants to this complex world of genomics.
I know, and it makes me wonder, like, if we can move past these simple ideas of what a gene is, what new ways can we think about how genes and the environment and just chance all come together to shape life?
That's a great question, and I think it's one that scientists and philosophers will be wrestling with for a long time to come.
Well, on that note, I want to thank you so much for joining us on this deep dive. It's been a fascinating journey.
It really has.
And for all of you listening out there, we hope you've enjoyed exploring the world of the gene with us.